This conference will now be recorded.
Hello, everyone, and welcome to the Invasive Mussel Collaborative webinar, Invasive Mussel Genomics, Innovations for Control Methods. My name is Erica Jensen, and I'm with the Great Lakes Commission. And on behalf of the Invasive Mussel Collaborative, thank you for participating in today's webinar. For those of you who are not familiar with the collaborative, the Invasive Mussel Collaborative was convened in 2015 to identify opportunities to advance scientifically sound technology for management and control of invasive Dreissenid mussels in order to produce measurable ecological and economic benefits. If you'd like to know more about the collaborative, we encourage you to visit the website at www.invasivemusselcollaborative.net. Today's webinar is part of an ongoing series hosted by the collaborative. The purpose of these webinars is to facilitate learning and information sharing on topics of common interest within the invasive mussel community. It is also the first webinar in a mini-series on invasive mussel genomics, and it will be recorded and archived along with past and future webinars on our website. Today we will cover the U.S. Bureau of Reclamation's prize competition, Eradication of Invasive Mussels in Open Water. The first prize awarded in this competition proposed a novel genetic modification-based method for control, and we're pleased to have speakers today from both the U.S. Bureau of Reclamation and Biomilab joining us today. While we always strive to minimize technological issues during these webinars, we recognize that sometimes issues may arise, and we apologize for any problems you may have. And on that note, I'd like to cover a few logistical items before we get to our presentations. Audio for the webinar can be streamed through your computer's speakers or through a telephone. To use the phone line, select the phone call option from your GoToMeeting dashboard to access the number to call into, as well as the conference ID number and audio pin to connect to the audio. To use your computer's audio, please select that option instead. If you are experiencing technical difficulties, you may send a message through the chat box at the bottom of the GoToMeeting dashboard. And in order to minimize the potential for interruptions and background noise, we are automatically muting all attendees, and attendees will remain muted throughout the webinar. That being said, we do encourage and will accept questions from attendees, and we encourage you to submit those through the chat box in writing. Questions may be submitted at any time during the webinar, and depending on time, we will get to the questions after each presentation, or we will cover them after all the presentations have been given. We'll do our best to take all questions and comments during the webinar, but if you need to leave before we've gotten to your question, we encourage you to review the webinar recording that will be posted on our website to hear your question and answer. So those are all the logistical and housekeeping items I have to cover. So at this point, I'm happy to introduce our first presenter of today. Our first presenter is Sherri Pucherelli. Sherri is a biologist and invasive species research coordinator with the Bureau of Reclamation within the Department of Interior, and her research has focused on invasive mussel control at hydropower facilities and mussel monitoring. So Sherri, we are giving you control, and hopefully you are off mute and are able to start your presentation.
Hi, Erica. Can you hear me and see my presentation okay?
Yes, we can.
Great. Thank you. So today I'll be presenting on the Bureau of Reclamation's prize competition program, and then also some more about our mussel prize competition, what the results were, and how we ran it, what our next steps are planned to be. All right. So prize competitions are great. They allow access to a previously untapped resource of knowledge, a knowledge base. So here at Reclamation, we're always dealing with tough problems, just like any other agency, where experts within Reclamation and our collaborators outside of Reclamation are working to solve these problems, but haven't been able to come up with a solution, or the solutions are too expensive or not practical to actually implement. So when we do these prize competitions, we put this tough problem out to the general public, and we're able to reach people that we never really knew existed, people in completely different areas of study that have unique perspective on the problem and are able to bring their experience and help us solve this problem. So our prize competition program at Reclamation is run out of our research and development office. We've launched 19 competitions since 2015. We actually just recently launched 2 in the last 2 weeks, and we have 3 additional competitions planned to launch in 2019. The authority we have to run these competitions comes through the America Competes Reauthorization Act of 2010, and all federal agencies are able to run prize competitions underneath this authority. At Reclamation, we are running prize competitions in 3 main topic areas. Those are water availability, infrastructure sustainability, and environmental compliance. So although we are the ones who are here at Reclamation that are coming up with our ideas and our problems for these challenges, collaboration is really vital. It's important that we are working with other federal, state, and local government agencies, water districts, private industry, academic partners to develop these challenges, and then also to have experts outside of Reclamation to accurately judge the solutions that we receive. So our Mussel Prize Challenge was, we had help administering that with a company called Innocentive, and they post the challenge for us. They help us deal with the solutions that we receive, and they just help manage the challenge. In 2018, Reclamation entered into agreement with NASA's Center of Excellence for Collaborative Innovation, and this just allows Reclamation access to additional vendors beyond Innocentive, and these vendors kind of compete for our prize challenges. They're diverse in their specialties, and they just will help us have access to a broader solver community. And also through this route, we will be able to receive solutions from international solvers. For our Mussel Prize competition, we were only able to receive solutions from solvers within the United States. So now we'll move on to a little bit more about our Mussel Prize competition. So our objective here was to seek theoretical, innovative solutions to eradicate invasive zebra and quagga mussels from open water. And when we say open water, we're talking about large reservoirs, lakes, and rivers. And, you know, this is obviously a huge ask. We were really asking a lot here. Some of the specific information we wanted from these solutions were solutions that were very species specific, didn't impact other organisms in the environment, solutions that were cost effective, you know, able to effectively apply these to large areas in a cost effective manner, and then solutions that had minimal impact to the ecosystem and were also scalable to large water bodies. So we're talking about large waters like Lake Mead. So the prize purse we had for this competition, we had $100,000 in cash, and the competition was posted at the Innocentive Challenge Center website at their water pavilion. It was also posted on the U.S. federal government challenge platform at www.challenge.gov. And so the challenge was open between December 2017 to February 2018, and it allowed a 76-day submission period. During that submission period, 238 solvers registered for the challenge, and we received over 100 submissions. So Innocentive took a look at those submissions. They removed any one-word answers or solutions that were not related at all, and they provided us with 67 solutions to be judged. Our design team and judges panel consisted of 10 experts from both the Bureau of Reclamation, Army Corps of Engineers, USGS, Malloy and Associates, and Portland State University. And our judges panel, out of those 67 solutions, selected 3 winning solutions. 2 of those received a partial prize of $10,000 each, and then one solution received a full prize of $80,000. So the partial prize winners, they provided a really unique, well-written idea, but there were aspects of their solution that did not meet all of the criteria of the challenge, so we were only able to win a partial prize in that case. We finally had a press release announcing the winning solutions in October of 2018. So a little bit more about the winning solutions. One of the partial prize winners was Wen Chen, a research scientist at Harvard Medical School. And his idea was interruption of muscle settlement by targeting DOPA or foot proteins using aptamers. And the use of aptamers here was really the unique idea to inhibit settlement. This was a really well-written proposal. We did see some limitations, though, that prevented us from awarding full prize. We believe that the production and application of the aptamers could potentially be cost prohibitive and not scalable, which were two main important criteria of the challenge. Also, we were kind of concerned that the aptamers would be degraded in an open water environment. But, as I said before, these were theoretical ideas. So, it was a unique idea. Our judges panel didn't see that it could be awarded a full prize, but we do hope that they continue the research and are able to have some success with this. The other partial prize winners were Abzar alum and Stephanie Bone of Biotech. And their idea was to use genome modification to develop male mussels with sperm that contain an optogenetic switch. So this switch is triggered by the light, and it drives a cyclin B expression. And so with that cyclin B expression, it can result in the death of the fertilized egg. So the use of that optogenetic switch was kind of really the novel idea that we saw with this solution. We were concerned, though, that if the cyclin B expression is lethal and the fertilized egg dies, the modification wouldn't be passed through the population. And this could just be concerning when you're trying to treat large populations, large water bodies. It could potentially increase the cost of something like this if you have to retreat. Also, mussels settle in locations with low light penetrance may not be impacted. That optogenetic switch might not be triggered. But again, a great solution that we received, and it does have a lot of potential. And so on to the full prize winners, Steve Suhr and Marie-Claude Senut of Biomilab. And they're going to be the second presenters on today's webinar. Their idea for the prize challenge was to utilize CRISPR-Cas9 mediated genome modification to induce a lethal cancer in mussels. And this cancer is malignant hemic neoplasia, and it's a pretty unique type of cancer that can be transferred from one mussel to another by proximity. So basically, it's like a contagious cancer. We thought this was a really well-written and complete solution, very novel idea for the treatment of mussels, and we thought that the solution met most of the criteria for the challenge, so we were able to go ahead and award a full prize here. So once we awarded the winning solutions, here at Reclamation, we have multiple options in pursuing these winning solutions. So we could either run a phase 2 and 3 prize competition, and in this case, we would have opened up another competition to the winners, and then also again to the general public, where they could take their theoretical ideas, develop them so that they have something to actually test at a lab scale setting. We could also go another route where we just decide to do reclamation-led research, where we work directly with the winning solver to complete the research. We could go with technology transfer, or we could just decide that we don't want any further involvement in the solution. So what we've decided to do is move forward with reclamation-led research, working directly with Biomilab. And we're working on developing a cooperative agreement with them currently to continue this research. The solvers were interested in working directly with us, and they have experience in the lab space to do the work, and they're actually able to devote multiple years to do the research. So that made it kind of played into our decision in going forward with the cooperative agreement. Overall, we were pretty happy with the results of the prize challenge. I was a bit skeptical when we started because we were asking for so much, but we did receive quite a few interesting and exciting solutions, so I think it was really worth it in the end. So on this page, I have my contact information if you think of additional questions. And I also have Jennifer Beardsley's contact information. She's Reclamation's prize competition coordinator, and she can answer any general questions about prize challenges. And then this website here on the bottom is the location where you can find more information about Reclamation's prize challenges that we have run in the past and we're running in the future. So Erica, should I take questions now or should I wait till the end?
Thanks, Sherri. It looks like we don't have any questions yet, but I would encourage folks, as I mentioned at the beginning, to type those questions into the chat box. Sherri, I think just stay tuned and we'll let you know if we get any questions at the end.
All right, thank you.
Thank you. So our next presenter today is Steve Suhr. Steve obtained his Bachelor of Arts degrees at the University of Kansas and his PhD in biochemistry and molecular biology at Northwestern University. He has been a research fellow in the laboratory of Dr. Fred Gage at the Salk Institute, the laboratory of Dr. Daniel Goldman at the University of Michigan, and Dr. Joseph Sibeli at Michigan State University. He was a research assistant professor in the Department of Animal Science at Michigan State and continues at MSU as an adjunct faculty. Marie-Claude Senut earned her PhD in molecular and cellular developmental biology at the University Pierre et Marie Curie and subsequently held a tenured research position in the laboratory of Dr. Jean-Marie Besson in Paris, France. She assumed research associate positions in the laboratory of Dr. Fred Gage at the Salk Institute and the laboratory of Dr. Daniel Goldman at U of M and the laboratories of Drs. Arshad Majid and Jose Sibeli at Michigan State University and the laboratory of Dr. Douglas Rudin at Wayne State University. She currently is MSU adjunct faculty in the Department of Animal Science. And Dr. Suhr and Senut left academia in 2014 to found Biomilab, LLC, a hybrid business that combines the fee-for-service function of a contract research organization with in-house R&D aimed at discovering and developing new innovative research tools, reagents, and methods to advance the biosciences, medicine, and ag bio. So thank you both for being here. You should have presentation control, and hopefully you're off mute and ready to go.
Okay, we just off muted. Can you hear us?
Yes, we can. And
okay,
we can see your presentation.
All right, super. So we are Steve Suhr and Marie-Claude Senut, and we are both basically Biomilab. We work primarily with academic clients, helping laboratories that may not have enough postdocs or maybe switching to a different kind of science. We like to jump in and help them however we can to forward their science. In addition to our work with academic groups, we also have a little bit of in-house R&D, and this project was basically started as an in-house project that we thought we'd take a crack at. And so the title is Eradication of Invasive Quagga and Zebra Mussels using Engineered Disseminated Neoplasia. And as Sherri pointed out or mentioned, this is a very unusual and rare form of cancer that is, okay, hold on a second, I'm seeing if I can advance. Cecilia, do you see slides going forward?
Not yet. You may need to go look in your lower left-hand corner of the slide. There should be an arrow forward button down there.
Let me see.
Oops, no, just to the left of the marker, I think.
Okay. Oh yeah, here, here. Aha!
There you
Now I got it. So what is disseminated neoplasia, which we're just going to call DN? So disseminated neoplasia, it's a very rare form of cancer where neoplastic cells are spread by direct transfer from one individual to another. It's very rare. There are really only a couple of disseminated neoplasias that are known in nature. One of them is canine transmissible venereal tumor, which I won't show you any pictures of in a webinar. But it's an amazing cancer actually, because it was recently found that the tumor that is associated with CTVT originated in an animal that lived about 11,000 years ago. So by at the time when this dog lived, there were still giant sloths and mastodons walking the earth, and so this is really an ancient cancer that started in one animal and has now gone to hundreds of thousands and millions of animals over time and exists on all continents. Let's see. There is another fairly well-known disseminated neoplasia, and that is in fact in Tasmanian devil. And Tasmanian devil facial tumor disease was first really observed in 1996 up in a corner of Tasmania, here where this tiny blue dot is. And since that time, it has spread essentially throughout the entire island. It has resulted in at least a 90% decline in the devil population since 1996. The current estimates are closer to 95%, and it's projected that it would cause complete extinction of Tasmanian devils by 2026. And so efforts are currently underway to remove devils to outside places where they can be kept until the disease runs its course on the island. One interesting fun fact is that Tasmania is approximately the size of Lake Michigan in terms of square footage. So we might be looking at something similar to Tasmania when we're talking about trying to eliminate mussels. So there is one more form of neoplasia, of transmissible neoplasia that's known, and that's bivalve transmissible neoplasia that's only been really described in like the last 5 or 6 years. And this is, of course, what gave us the idea for our submission, because we had been looking into a variety of different types of CRISPR-Cas9 or genetically transmitted methods of suppressing the mussel populations, and we came across these reports of disseminated neoplasia, and it just sort of seemed to be staring us in the face that perhaps we could take that natural disease of bivalves and instead of doing the research that everyone else is doing on it, which is trying to prevent it, particularly in cultured mussels and other types of bivalves, if we could actually try to cause it in invasive mussel cells and then use it as a tool to suppress them. I just wanted to, these images here, this is showing these cells in Mediterranean mussel gonad, and this is showing the hemic cells, the normal and then the transformed or neoplastic version of those same cells that are growing uncontrollably. And so we've essentially decided that our strategy will be to try to take normal cells from invasive mussels, convert them into leukemic cells or disseminated neoplasia cells, and then reintroduce them in the population to create chronic disease. Oh, one thing I just wanted to mention in this comparison that came from a Metzger and Goff review was that one reason why these cancers are so rare is that it's very unusual for a cancer cell to be able to move from one individual, or even a non-cancer cell, any type of cell really, to move from one individual to another. And so in the canine version of disseminated neoplasia, there's been downregulation of the major histocompatibility complex related proteins so that this can move from animal to animal. That's also found somewhat in the devil facial tumor. But in addition to MSC downregulation, the cancer is helped because since Tasmanian devils are a very limited population on only a single place on Earth, essentially, they are also very highly inbred and very highly related to each other. In the case of bivalves, they're invertebrates and don't have MHC. So there might be some advantages and reason to think that we might be able to be successful at doing this in bivalves where we would maybe not have a chance with other types of organisms. So if we just picture some invasive mussels happily living on the bottom of on a river bottom or a lake bottom, then the idea here is that essentially we're going to transplant some of these disseminated neoplasia cells onto a target population in a target waterway. It will then engraft into one or more animals and become systemic in those as systemic cancer. And the hope is that essentially that systemic cancer will then spill out into neighboring cells, just as it does in the wild forms of this disease, and eventually take root in the population as a whole, and then at that point become basically endemic and kill mussels and continually affect all the mussels that are essentially in close proximity, and if the populations are large enough, perhaps also travel via water currents to other populations within the waterway. So we think that our hope is that we'll be able to essentially inoculate a very tiny population and then have the disseminated the neoplasia disseminate essentially to the rest of the population. So there are a lot of advantages to this, but a few of the ones that we think are the most important is that like a single inoculum could actually create an endemic disease that goes throughout the waterway and really causes collapse of the invasive mussel population. We think that there is essentially no chance of toxicity or injury to other aquatic or terrestrial organisms, with the exception, though, of very closely related species, like within other dry-sinid mussels. But by the same token, we think that it's very possible that we could develop a disseminated neoplasia that would act on both zebra and quagga mussels. Another thing that might play to our advantage is that the U.S. invasive mussel population is thought to be very highly inbred. Everybody's very closely related to each other and started from a small initial population. So they might be good targets for allografts like disseminated neoplasia. In other words, that they may be all so closely related that they would be highly tolerant of cells from one individual to another. Then some of the disadvantages of this is that we still don't really know a great deal about the molecular and cellular biology of dry-sinid mussels. And so we're really counting on our collaborators at USBR to help us particularly address a lot of the molecular biology that we're going to need to know to complete the project. Another thing is that it might be a relatively slow process. Population collapse may take a number of years to actually be complete within especially a large waterway. And then it's also possible that closely related species could be impacted. However, it seems that in the natural forms of this disease that it's limited to within species, but it doesn't extend to other family. So in other words, you might get within other dry-sinid mussels, but probably not to unionid or to anything else, a true mussel species or other types of bivalves. But that remains to be tested in a laboratory setting, basically. So the nuts and bolts of creating a disseminated neoplasia, there are a lot, and I'm kind of showing you this, not to go through it. We're only going to talk about one tiny piece of it that I just thought might be interesting to folks and that you might not have a lot of familiarity with, and therefore it might have extra interest to you. The very last point here is move everything outside. To us, this is the fairly straightforward part, and then when everything goes out into open waters where things might get really complicated. So the nut or bolt that we're going to tell you about in a little bit more detail is about the cellular events and factors that actually lead to carcinogenesis, because we're going to be attempting to make normal cells of an organism, and in this case an invasive mussel, into cancer cells. And there are shared, it is believed by all animal species, from man to mussel, that you are likely going to have to address a few components or a few factors that are involved in cancer in order to achieve a neoplastic cell. One thing you're going to need is immortalization. So you're going to need a way to actually make it so that that cell has the capacity to undergo cell cycle and divide forever without any damage, or without something that's going to force it to die after it normally should have reached a senescent state. You're also going to need something that pushes that cell to continue in the cell cycle. So you have to deregulate that so that normal cues that the cell would receive to stop dividing can be overcome. And then a lot of cells, when they are forced to divide and they pass their normal lifespan, they undergo programmed cell death. There are kind of safety backup measures in the cell to try to stop it from growing uncontrollably. And then there are also factors that are present in the environment that try to drive the cell to divide. And so we have to, in the absence of those in a cancer cell, we have to trick the cell into keep dividing, even when those things that it normally would be looking for and those cues to divide are not present. In human cancer, this is sort of I found this in this review article in Yale Journal of Biological Medicine, which has sort of the succinct steps and the key players in converting a normal human cell into a tumor cell. And one of those factors is TERT, which is involved in the immortalization of the cell, P53, which involves cell cycle deregulation and apoptosis, and then another factor that's been known for decades now that is a key that has taught us a lot about cancer is SV40 large T antigen, which is an oncogene carried on a virus that has the properties of being able to, by itself, convert a normal cell into a tumor cell. And so we're kind of proposing to use these guys, perturb these guys in order to try to convert a muscle cell into a cancer cell. So TERT, or telomerase reverse transcriptase, is a protein that's involved in the immortalization process and controlling how long the cell can live. We need to make a lot of that. We need to make more of it, or we need to make sure that it continues to be expressed so that the cancer cell we make is essentially able to continue to divide forever. We have to inactivate, or we're expecting to inactivate the protein P53, which is critical to controlling cell cycle, and also in the cell death that's associated with that. And then also SV40 large T antigen, like I said, has been known for decades, since the 60s, I believe, mid-60s. And this protein also, if we overexpress it, we can use that to convert cells into a neoplastic type phenotype. So just to quickly go over these guys, TERT, or telomerase reverse transcriptase, is a protein that basically puts the telomere bumpers on chromosomes, and that protects them from the rigors of replication. And this is a picture that was from the Sherri lab. You can see beautiful. This was using what they call FISH, fluorescent in situ hybridization, to essentially illuminate the telomeres that are on the ends of these chromosomes. And so these are what essentially protect the chromosomes from damage as the cell undergoes division. In mammals and birds and other organisms, telomeres are created very early in development. In humans and mammals are created essentially when you're a little ball of embryonic stem cells. And that's when your telomeres are created at their full length. And then when you begin to differentiate to actually form your somatic tissues, TERT is turned off completely. And this leads eventually to aging and the senescence of the cell, because every time it undergoes a replication, those telomeres are degraded just a little bit. But the trade-off here is that if a cell goes rogue and begins to divide too much, then these will be lost, and that will protect against cancer, because once the telomeres are gone and the chromosome is sufficiently damaged, the cell is going to die. So that's sort of something we've evolved with as a protection against cancer at the trade-off that we actually will undergo cellular aging. And I've worked with TERT and telomerase and telomeres previously, say in a project where we reprogrammed human fibroblastic cells to an ES cell-like cell using a cocktail of gene factors. And as you can see in the adult cells, that you have very tiny telomeres that are visible on chromosomes as these tiny red dots, and that these become a lot beefier and chunkier when the cell is made into an embryonic stem cell, and that's visible here. So this is kind of just to show one example of how we've used TERT in the past or analyzed TERT. There is one interesting connection of TERT to aquatic organisms. And in aquatic organisms like teleost fishes, which we've worked with before, and some other species, TERT expression continues really for all of the life of that animal. And so we've also had some interest in controlling carp using genetic modification, invasive carp species. And some of you may not be aware, but and unfortunately I don't remember the specifics, but the oldest carp known, I think, is something like 230 or 240 years old, and one was kept from generation to generation in one Japanese family. Also, when I was sort of looking at aquatic organisms and TERT and telomeres, I actually found this article from USA Today, which is saying that scientists accidentally killed the world's oldest animal. Well, they actually froze a whole bunch of these clams, and it did turn out, though, when they actually counted the rings, and I apologize to everybody in this that is actually a bivalve scientist, but to my mind, they counted the rings in this clam and determined that it was, in fact, 507 years old. So it's likely in bivalves and mollusks, at least some, that they probably express TERT for their entire lifetimes and are able to maintain telomere integrity almost indefinitely. But it remains to be seen, and it remains to also be seen what TERT does in the Dreissenid mussels that we're discussing. So the second factor that we're interested in manipulating is tumor protein P53. It's referred to as the guardian of the genome, and it is the protein that's most frequently mutated in human cancer, and that's probably the main reason that P53 has been studied so extensively now for at least 20 years and probably closer to 30. So if a cell lacks P53, then that disables its capacity for cell cycle arrest. And also, interestingly, P53 mutation has been associated with the bivalve disseminated neoplasia. It's not a causative, it hasn't really had that link, but it has been found. P53 mutation has been found in some of the few bivalve disseminated neoplasias that have been found. So we think there's a good chance that mutating P53 is also going to help us get a lot closer to disseminated neoplasia. I just want to show this because it's such a dramatic example of P53, what P53 can do in the context of an organism and also in causing cancer. These are in mice where a knockout was created in one allele of P53, leading to heterozygotes. Those heterozygotes were then mated, giving rise to a population of heterozygous mice with two very perfect copies of P53, and mice with two completely defective copies of P53. This is essentially a kill curve. And so if we look at the age in weeks of these mice when they died, whether of cancer or some other cause. In the animals that had 2 intact copies of P53, roughly 50% of the animals lived up to 120 weeks with only a single functional copy of P53. That's reduced down to 75 to 80 weeks. And in the case of animals where both alleles of P53 are knocked out, half the animals are dead by roughly 20 weeks. And all of the animals that are dying in the, particularly in the no P53 version, they're all dying of tumor formation, cancer. So P53 is really a key operator in the carcinogenesis of a variety of animals, and it's likely to also be involved in muscle. So the last one that we also want to test, and we have a few other guys too that we might look at, but these are our 3 primary proteins of primary interest, is large T antigen. It's derived from SV40, was discovered in the 1960s, a contaminant of poliovirus or something. Polyomavirus, large T antigen is a polyomavirus, and the polyomavirus needs S phase in the cell to replicate, and so basically T antigen forces infected host cells to undergo continuous S phase to essentially become a cancer cell by interacting with P53 and RB and some other internal proteins. And a temperature-sensitive form of T antigen was isolated a long time ago now by Jett and Sharp, and I have used that version of T antigen many times in my own research. And this is just an example of how that works. So if we place T antigen into satellite cells, skeletal muscle satellite cells, which are the cell that is the progenitor of skeletal muscle myofibers, normally these cells just sit and wait until muscle damage occurs. Then they undergo a couple of divisions to produce a little bit more of themselves. And then they fuse into the existing multicellular myofibers. If I put T antigen, temperature-sensitive T antigen into those satellite cells, then I can essentially hold them in a perpetually dividing state, basically a cancer state, where they will continue to grow forever at 33 degrees. If I shift them to the non-permissive temperature, and in this case I was actually controlling the T antigen with a couple of factors, temperature and another drug called doxycycline. If I did those 2 changes, then the satellite cells would essentially all differentiate and resume their normal state and fuse into multicellular myofibers. So this is just a demonstration of how T antigen can essentially dominate the cell cycle and force that cell to continue replicating and also continue as an immortal cell. So with both of these factors, and I unfortunately cannot go into this in great detail because it would just take too long, but so we essentially need to manipulate the levels of these oncogenic factors. So we need to overexpress TERT and T antigen, and this would probably be done via transgenesis, where we would introduce TERT and T antigen into the cells with a plasmid construct or a virus. And then we need to knock out P53. We need to get rid of its activity, and we would probably do this using the method of CRISPR-Cas9. And so these are just examples of times in the past where we have introduced novel genetic material into cells to, say, study Huntington's disease, to reprogram fibroblastic cells into pluripotent stem cells, or say to make reporter animals, like this is a transgenic zebrafish that we created in Dan Goldman's lab at University of Michigan for analysis that we were doing there. So we have quite a bit of experience in generating recombinant DNA constructs and moving them into animals and cell types in order to do analysis. Then for the knockout, we would be using the CRISPR-Cas9 gene targeting, and both of these things could basically make a webinar all on their own. But essentially, the Cas9 method uses the protein Cas9 complexed with what's known as a guide RNA. It's not quite as simple as I'm making it out to be here, but that would then go in and recognize a sequence in the genome of the cells, of the hemic cells that we would isolate. It would then use that guide RNA to target our specific target, in this case P53, and then introduce a mutation into that gene. And hopefully we would hit both alleles. I just wanted to show over here is just an example we have done recently for a client. There's too much here to talk about in the time that we have. But if we look down here in panel C, if we essentially, we're trying to target a gene in the canine genome, and if we introduce different guide RNAs, here called T1, T2, T3, and T4, we can kind of assess how well we've actually done that mutation. If we see these two bands on the lower part of the gel here, then that means we targeted nothing. The stronger this is, the less that was targeted. The more that's up here in this position, where we've knocked out a local site very close to our targeting guide RNAs, the stronger that these bands are up here relative to the bands down here is the amount of targeting we're doing in the cells that we have in our culture dish. And so, as you can kind of see from this, we can usually find conditions and combinations of guide RNAs that will allow us to target a fairly high percentage of the total alleles in that cell. So we believe that there's a good chance that we will be able to, in a few cells, hit both alleles of P53 and knock out its capacity to function, and thereby providing us with essentially a cancer cell made from a wild type cell. There's also advantages that we'll have to that, and the cancer cells will proliferate and stand out relative to the non-modified cells. But again, that will have to be for a future webinar. So in conclusion, I just want to say that, again, that disseminated neoplasia is a very rare type of cancer, and it's transmissible within only really closely related species. There are forms of disseminated neoplasia that have been found in the wild, but also in cultivated bivalves, and so that's why it's been of interest to the scientific community in the past. The molecular pathways of oncogenesis are pretty well understood, and they're applicable to a variety of different animal species, and so we think these can probably be exploited to induce disseminated neoplasia in the Senut mussels. And one thing that is going to be necessary as part of this project is that we're going to need additional understanding of the molecular and cellular biology of these mussels so that we can design and refine the methods by which we are producing the engineered disseminated neoplasia cells. And last of all, I just want to say thanks to the folks at USBR and the organizers of the Invasive Mussel Challenge, and our scientific colleagues so far that we've interacted with at USBR, Sherri and Jackie in Yale. And then this kind of also had a genesis in some local people, Aaron Purcell at MSU, Ashley Balder-Jelgen, who's at NOAA, Seth Herbst, Tom Gagne, they're at Michigan DNR, Donna Cashian at Wayne State. All these people have in some way kind of contributed to varying degrees to the genesis of the project. And then I finally just wanted to say thanks to the webinar organizers and the Invasive Mussel Collaborative, and we're happy to be hopefully part of you going forward. Thanks very much.
Thank you, Dr. Suhr. We do have a few questions that have come in, and we do have one for Sherri as well. Sherri, if you want to take yourself off mute, but the first question I'll ask will be for Dr. Suhr, and that is there current evidence about transmissibility between mussel species, or is DN considered to be species specific?
Okay, from what little is known, and there's not a great deal known out there, in all the mammals that it has occurred in, it is incredibly species specific, mostly because of the immune response, the major histocompatibility response that we have, where you cannot exchange cells between individuals and have them hope to survive. In the case, though, of bivalves, since they have a less sophisticated immune surveillance, we think it's going to be able to probably, and the evidence suggests that it can go cross species within very closely related species. There so far is nothing documented that is crossing family. So I believe that there's like I'm sorry, I don't know these real well, but there's a carpet clam and another clam that has a very similar name. There have been cells, neoplastic cells, that have been found crossing from those two organisms. But I believe that in mussel and the other bivalves, there probably just aren't enough examples known to know if it can happen. But I think it is believed that because every organism sort of has its own metabolism and its own preferences, then it's highly unlikely that the cells will travel across families like that. But that's something we have to investigate. That's absolutely something that has to be investigated in a laboratory setting.
Thanks. And so we had a related question about the possibility of unionid mussels being affected. So I don't know if you want to say any more or feel like you covered it with that response.
Well, I don't know much about them, but I do think that they are, I'm not positive, I think they are a true mussel. I'm guessing they are a completely different family from the Dreissenidae. I think the ones that we would have to worry about the most are probably Dreissenidae. And I don't believe there are a lot of Dreissenidae in North America, or I even read, I believe that the only Dreissenidae in all of UK is the invasive mussel population. But that's unfortunately not my area of expertise.
Erica, this is Sherri.
Yes.
I have a couple of the collaborators on this project in the room. If we can respond to the question, this is Yale Pasabanek.
Hi, this is Yale. Just to follow up on that question, I would say that we consider that the transmission to unionids would be highly unlikely given their divergence between them and Dreissenids, but that's speculative. I think, as with any environmental treatment, especially when it comes to any kind of genetic manipulation, there would be, after the hopeful demonstration of efficacy of these approaches, there would just be a tremendous amount of very controlled laboratory testing before there was even the thought of some kind of introduction in the wild. So that and anything else in the system would be challenged with these cells to see if there was any impact whatsoever over a long time period.
We agree totally.
Mm-hmm. Great. Anything else anyone wants to say on this topic before we move to the next question?
Nope, I think we're okay.
All right, thank you. So the next question we have is, Dreissenid mussel population genomics shows, like other aquatic invasive species, that there's a high genetic diversity in the invaded range. So the devil disease is believed to be promoted by host inbreeding. How about the canid version of transmissible neoplasia?
Okay, this I think is going to be best answered by USBR folks, because we've actually discussed this before. We brought up the possibility that we could raise cells from mussels taken from multiple sources, but I think Yale and Jackie can probably comment on this, that there's a fair amount of evidence that there's a lot of homogeneity in that population, but I'm going to let them pick that up.
Hi, this is Yale again, and Mike McCartney and I carried on a little bit of a conversation on the sidebar already. Our preliminary data from some microsatellite work that we did in collaboration with the U.S. Army Corps of Engineers suggested that there's comparatively low population diversity in quagga mussel populations in the lower Colorado River system. The resolution with that was relatively small. We had a handful of markers, and so it's definitely, as we're developing the genome sequence and genomic tools, it's something that we're going to revisit and interrogate more intensively. Our working hypothesis has been that since this is a second introduction after introduction of the Great Lakes, and it was probably very small founder populations brought in likely on recreational boats, that there may have actually been a secondary population bottleneck in the introduction into these Western United States systems, and that if we are indeed find that the population diversity is low, that would account for it. I won't get too much into our genomic data yet, but we are seeing some diversity in our initial analyses, our genomic stuff, so it's definitely going to have to be revisited more in depth and with more tools. So.
Yeah, also, this is Steve at Biomilab. We've also considered that it almost may be also in a way like a flu vaccine, sort of, where we might combine the most prominent strains of cells together and then use them as a common inoculant, or depending on what we sort of find out from the biology, it might be that also we would have a pure inoculants of a specific cell, and then those would work best with certain populations, and it would just have to be empirically tested.
Great, thank you both.
Sure.
I'm going to move on to the next question, unless you wanted to add something.
Nope.
All right. So taking a slightly different tack with this question, how are the mussels infected? Are individual mussels extracted, infected, and then thrown back into the waterway, or would it be done through a chemical addition in the waterway?
Well, from our end, we're envisioning, possibly both. We may infect mussels in the lab and then take them out into the field and essentially transplant them into the colony. Or I think there will probably be ways for us to actually inoculate in the water itself. And one thing we would probably look for in the long run are carriers, where we might be able to sort of cause the cells to fall nicely onto the target. And that would, of course, involve folks in wetsuits with tiny droppers of some kind swimming around in the water. But I still think it's possible. And then if it does work well, and if the cells are fairly efficient, then it could be deployed as some sort of gel or something that would then fall down onto target cells. But we could envision either type of approach.
Thank you.
Sure.
We have a couple more questions, so I'm going to keep rolling along. As the target population gets smaller and smaller, presumably the remaining individuals would be spread further apart. Do you think the proximity required for this technique might make it effective at knocking down populations but be prohibitive to extirpation?
I'm not sure I caught the very end of that, but that is definitely true. When the population begins to get small enough, then it might be harder. It might almost become like a boutique deployment of the reagent. We don't. We'll just have to cross that bridge when we come to it.
All right. Next question. It seems to be relatively well understood how to make cancer or immortalized cells. But as you say, the disseminated neoplasia is a very rare form of cancer. How well understood are the mechanisms that allow the cancer cells to survive outside the host and then engraft onto a non-self individual? Would this research lead to the generation of many cancer cell lines that are unsuitable for the desired type of control?
I think that I'm afraid I still have a lot of reading to do on disseminated neoplasia in general. But from what I understand so far, I don't believe that very much is actually known about the mechanism of exchange. And one thing I do remember reading, I think is on that table that I had in the 4th or 5th slide of the talk, where they're talking about the route of transmission. It's easy to see how it's happening in the case of the canine venereal tumor, and even in the case of the devil. But they just say, presumably through water for the disseminated neoplasia in bivalves. So that's an open question, and I think that the answer is probably going to be maybe very interesting science in that. I'm hoping to get a chance to have some sort of communication with some of the folks out there who are experts in this, so that we might better be able to understand that. Another thing is that my guess is that this hasn't been experimentally reproduced in a laboratory setting very often, if ever. And so we might be able to get some basic biology out of these things, and we might actually learn quite a bit about mussels and the exchange of tissues. If it's actually true that mussels or other bivalves are sort of amenable to an exchange of materials like this, it would be very interesting if there were was good reasons for tissues to exchange. I don't know. I think it'll be interesting. Well, one thing we've thought about is of course, we're also probably going to try to mark a lot of our cells with some sort of detectable transgene. And one thing we've thought about is using like luciferase assay, where we could even, this is getting into science fiction a little bit, but it would be so cool if we could actually use, like we could float luciferin into a population of mussels and actually observe the neoplasia moving out, almost like a radar image of a storm coming in or something like that. But one thing we would almost certainly do is probably try to harvest material from animals close to patient zero, and then move from that and see when we could detect cells that were definitely derived from the neoplasia. Hopefully we'll learn some about that.
Thanks. So this next question touches on a topic that came up earlier, but any of the presenters may or friends may want to expand upon a little bit. We have a question about how much testing would this go through to ensure that it cannot spread across the species barriers? And I'll expand that a little bit to maybe the Bureau of Rec folks, if you can speak a little bit more to the procedures and processes, if you can, that would be used to verify that.
You know, I'm not exactly sure that we know how much. We're going to be doing quite a bit of testing. That's kind of many years, I think, in the future. But it would all be lab scale testing, like Yale mentioned before.
And it would all be, part of regulatory agreements with other agencies as well. And so it would be a collaborative effort to determine that extensive testing is done and that it's to everyone's satisfaction. So.
Yeah, and this is Steve. We agree too. I mean that we're really at this point we're just we are keeping the focus exclusively on seeing if we can generate the reagent, seeing if we can get something to happen in a laboratory setting, and then after that we'll probably use agencies like USDR as our guide to how to move forward.
Thanks. Well, it looks like we're reaching the end here. We did have one process question, Sherri, related to the prize challenge, and that was why the challenge was limited to U.S.-based participants.
And yeah, unfortunately, it was limited to U.S. participants because of the company we were working with, Innocentive. I'm not exactly sure why, but they were limited to only receiving submissions from U.S. citizens. And that's why we've gone to that agreement with NASA now, where we can work with other companies that can accept those international submissions.
All right. Well, we're reaching the end here, so I want to thank all of our presenters and everybody who provided questions and responded to questions and participated in today's webinar. I did want to touch on something that I mentioned at the beginning, is that we will be holding a second webinar is part of a mini-series on this topic, specifically invasive mussel genomics and control and potential for control. So the second webinar, which announcement will go out for soon, will be later this spring, and it will review progress towards sequencing the zebra mussel genome and quagga mussel genome and potential implications for management. And we are working with one of our Invasive Mussel Collaborative members, Michael McCartney from the University of Minnesota, to coordinate that webinar. So I encourage all of you to participate in that when that happens. But otherwise, we are concluded for today. Again, thank you to everyone who participated, and I also want to acknowledge the U.S. Geological Survey and the Great Lakes Restoration Initiative for providing funding support for the collaborative. And as a reminder, we did record this webinar, and it will be posted on the collaborative website within the next day or two. And if you have any additional follow-up questions or comments, please feel free to contact us at the Great Lakes Commission, and if we can't answer them, we will find somebody who can. So thank you very much, everyone, and have a wonderful weekend.
Thank you.
Thank you.